Okay, hello everybody. This is Psych 3280, week 6, Neural Correlates of Consciousness. And this week we finally come to the before getting started for the week of the 6. Let's recap week 5, Neural Correlates of Consciousness. So first, we talked about the argument in support for color vision at the periphery. And there I presented several evidence. One of that is a brief presentation which doesn't allow the eye movement, still induces vivid color sensation in the periphery. So, we can remove eye movement artifacts like remembering or expecting what you have seen before with the phobia is filled in at the periphery at any moment. Okay. And then the second evidence was that the neurons at the high eccentricity have a bigger receptive fields so that it can collect color-related information effectively if the presented stimulus is uniform texture. And then the convergence effects, the higher level neurons are receiving a bigger extent of the neuronal input from lower visual areas, accumulates over the hierarchy. So at the B1, B1 can get the input from the retinal ganglion cells that cover multiple retinal ganglion cells. And then B2 or B4 neurons can even further combine the B1 neurons, which has a different location of the receptive fields, so that in the end, the higher the hierarchy a neuron lives in, it has a chance to accumulate information across over the wider areas. And therefore, uniform surface can potentially generate a strong activity in the peripheral neurons that have a bigger receptive field. And so I argue that the uniform colored surface can be optimum stimulus for such receptive fields. And this relates to the fact that, for example, when you go outside and then see the blue sky at the periphery, the blue can be quite vivid. It's if it's a very uniform blue. All right. On the other hand, I also presented argument against for the color vision at the periphery. And one is that the cone density, it's not the number, density, number per given areas of the retina decreases rapidly of the fovea. And in V1, the fovea is overrepresented at the expense of the periphery. So even though the peripheral neurons can have a potentially bigger, receptive field, the number of the neurons and also the dedicated amount of the resource for the periphery is in fact very, very small. And this fact called the cortical magnification implies that the per vision at the periphery. But this is something that not so many people realize, but this cortical magnification factor in the visual cortex is uniform across all the properties of the vision. So not only the color, but also the visual motion and also texture, as well as recognition of the object, all gets affected by this cortical magnification factor at the periphery. Okay. Next, and then we also went over the neural mechanism underlying blind sight, and 90% of visual inputs from the retina goes through the lateral geniculate nucleus (LGN) to primary visual cortex B1. And the rest of that also has, there are 8 more other pathways, but mainly goes to the superior colliculus, and the pulvinar to the cortex. And this imposes some kind of shortcuts and multiple pathways to from the eyes to the cortex. And the important thing is that the latter pathway, superior colliculus and the pulvinar pathway, can mediate eye movements and also localization in a non-conscious manner, the one that's observed in blind sight patient. Okay. And then along the way, to explain this peripheral vision, color vision, and also blind side mechanism, we encountered several important concepts and features of the neurons in the visual system. And that's going to be, again, reviewed in this week, but briefly, receptive fields and visual hierarchy. So the receptive field properties became more complicated, such as at the level of the retinal ganglion cells and the LGN, it's usually on-center or off-center, and onset response or offset response. So it's pretty much like, 4 types of the responses can cover quite a bit of the description of the retinal ganglion cells and the LGN. However, as you go to B1, this becomes not possible, and there are classes of the neuron that responds to oriented lines, and the different also direction of the motion also evokes a slightly different direction of the activity. And there are also neurons that are sensitive to the color and wavelengths and things like that. So it becomes more and more complicated, even from a retina to B1. And also the size of the receptive field, as I mentioned as a sort of the convergence rule, tends to become bigger as you go in the higher hierarchy. So V1 neurons have a bigger receptive field than the retinal ganglion cells or LGN. And also, but temporal latency-wise, it doesn't necessarily mean that the receptive field, the neurons at the higher visual hierarchy responds later than the lower hierarchy neurons. So some of the V1 neurons can respond actually later than the neurons in the prefrontal cortex. And we are going to cover some of these interesting neurons in the later lectures. Okay. And that also relates to sort of the relationship between the feed-forward pathway and the feedback, and how come the things that comes later, such as backward masking situation, erase the conscious perception of the things that came before. So that's the temporal hierarchy of the brain. And then also, each of the visual cortex, not only visual cortex, but across all the cortical areas, have an horizontally speaking columnar organization. So the receptive field properties change gradually and sometimes abruptly with a flip of the left to right, ocular dominance or direction preference and so on. There is some kind of columnar organization. Within vertical dimension, as you go from a top layer to the lower layer, usually neurons property, receptive field property changes gradually. And however, across the layers, what is different is that the connectivity pattern to and from other areas of the brain, as well as within the area of the brain. And this layer structure is, in fact, the driving factor for the Feldman and Van Nessen to derive this hierarchical map of the brain. Okay, so these are the important concepts of the visual system. And then multiple visual pathways to explain visual, blind side and other phenomena. Okay, so this week we will address the question of what is the link between neurons and phenomenology. What is the role of the V1 for conscious vision? So towards the end of the last week, I mentioned that all of these V1 topological features doesn't seem to match with what we perceive consciously. But this becomes really problematic if the V1 is actually responsible for conscious vision. If not, it doesn't matter so much. And then we also talk about the property of the visual cortex beyond the V1 for the rest of the lecture. Okay, so in terms of learning objectives of this week, you're going to be able to answer that what is a neural correlates of consciousness, NCC. So that's the concept needs to be clear. And then what are the positive and the negative evidence for V1 as the NCC. And then what are the properties of the neurons in the higher visual areas? And then, so these are the three main learning objectives of this week. And I put chapters 5, 6, 7, 8 as a reference chapter. And also, I also put Chris Frith's Trends in Cognitive Science paper as an optional reference. The reason becomes clearer as we go in the first part of the lecture this week, but it contains quite a useful framework for NCC search and empirical or empirical oriented question for consciousness study. So if you want to use that for your final assessment, for the project presentation, that would be a very useful kind of paper to read. Okay, so let's get started. In week 5, we encountered this figure of the symptoms and how I explained how it gets projected into the V1. It's flipped upside down on the left and right, and then also phobia gets magnified like this way. And then this raised the following fundamental questions, such as, why do we not see images upside down? And why do we not see the image in a distorted way? What is the relationship between the outside world, our experience, and the neurons in the brain? This may sound stupid kind of questions that you may reject outright, but it's not really trivial to say why, what to give a sort of the convincing explanation about all of them. In fact, if you think about animals or other types of creatures such as computer program, especially the robots, they can actually operate completely all these images projected, especially if you plug in some kind of cable to V1-like areas for the computer or robots, and then project it back to the screen as sort of the putative their conscious experience. It's totally plausible that the robot or other creature sees the world like this way, upside down and distorted, and then really weird kind of relationship between the outside world and its experience and the processing neuronal kind of architecture. As long as the other behavioral or report kind of mechanisms are consistent with all these topology, or the relationship between the neighborhood of visibility and outside world and also representation. In fact, we can prove that there is no mathematical reason why this is not the case. So, just if this is a very difficult kind of argument, you can just imagine and say that this is not a trivial kind of question, because in fact we can imagine the robot that works like this way, and that's enough for now. Okay, and in fact all of these kind of consideration made this question about consciousness very difficult, and notoriously difficult to the extent that some of the philosophers thought that this is a hard problem that it's impossible to tackle with the scientific questions. And on the other hand, leaving aside these kind of super fundamental questions, since 1990, I also mentioned in the week 1 lecture that an empirical scientific research program started. And that's the search for the neural correlates of consciousness. And this is primarily driven by my previous PhD supervisor, Koch, and previous Nobel laureate, Francis Crick. But what they basically asked was, rather than going into the kind of very difficult philosophical questions, they posed the following more tangible kind of question. So it seems, and they said that it seems probable, however, that at any one moment, some active neuronal processes correlate with consciousness, while others do not. And you can imagine that others would include prefrontal cortex or cerebellum from the lectures a few weeks back. And they say also that what are the difference between them? Them meaning that those neurons that correlate with the consciousness and those do not. Where in the brain are the neural correlates of consciousness? That's a more concrete question. And the correlates are much easier to deal with by science. And that's the one that we are going to focus on this week. Causal relationship from one event to the other, that's much more difficult to deal with. And also the philosophers also deal with or are interested in the questions such as what is the relationship, causal relationship between the mental to the matter. Something like this is very, very difficult questions, but as long as you confine yourself into the realm of the correlation, then you can make always empirical progress. Okay, so the NCC is defined as the following. Quest for Consciousness in glossary defines it as follows. First, that it's a minimal set of neural mechanisms or events jointly sufficient for specific conscious percept or experience. And this is what the book is about. Okay, so the point here that is important to note is the word minimal, and also jointly sufficient, and also a specific conscious percept or conscious experience. By this, what they wanted to get is the fact that, for example, retina is not the minimum set of the neural mechanism, and also it's not jointly sufficient. By that, what I mean is that, for example, I gave an example of the saccadic suppression in the last week. During saccadic suppression, your eyes move, and then you don't see what's going on outside of the vision during the saccade, and also preceding a little bit from the onset of the saccade. During this saccadic duration, retina's neurons actually do respond. However, recent studies seems to suggest that during the saccade, retinal neuron also starts to show some kind of depression in terms of the sensitivity. But that's taken aside. Anyway, so the things that's going on at the level of the retina does not get reflected in our conscious experience. If it is a case, then we should see much more a blank of conscious experience. As I emphasized last week, we do move the eyes 3 times second, and also each of the saccade is accompanied by 100 milliseconds of the sort of the blanking of the sensitivity, and that's not really what we are experiencing. So retina, for example, is not the minimally sufficient kind of the mechanism. And the same goes for the heart, for example. Heart can be replaced by some other mechanisms such as artificial heart pump, and still we can see and also hear and experience without any problem. And the other types of the mechanisms that are removed by this type of criterion would be like prefrontal cortex or cerebellum and so on. They do contribute to the normal functioning of the brain and normal functioning of the consciousness, but if you give qualification of for a specific conscious percept or experience, and if it's a jointly sufficient, then it's likely that these mechanisms are not the part of the NCC. Okay. And then, but this level of the definition of NCC has encountered lots of criticisms and further refinement as the science of consciousness proceeded, arrived at the following refinement of the NCC definition. So first is full of the neural correlates of consciousness, full NCC, and that's the neural substrate supporting experience in general, irrespective of its content. So, if the patient is awake and fully conscious, regardless of whether they are hearing or seeing, there should be neural correlates of consciousness that relates to their experience at that moment. That's a full NCC. The other one is a content-specific NCC, and that's that corresponds to the NCC I mentioned prior. The neural substrate supporting a particular content of experience, such as face, whether seen, dreamt, or imagined. As long as subject experiences, that content should be reflected by this content-specific NCC. Okay. So then the next empirical question is, how can we find the NCC? And as I mentioned in the beginning of this lecture, this paper, Frisch 1990 Trends in Cognitive Science paper, contains quite a good hints about how to find the NCC. And so I recommend you to skim through it at least, and then to employ it for your final project. Okay. So what they did in terms of conceptually is the clarification of the neural correlates into at least 3 different components. So first is the neural correlates of consciousness, and the other one is the neural correlates of sensory stimulation, NCS. And then another one is the neural correlates of the behaviors. So physical input comes into the brain through the eyes and LGN and so on. And then initially, there is a neural correlates of sensory stimulation that reflects what's out there, in terms of the processing. And then that leads to eventually neural correlates of consciousness that gives rise to our conscious experience. And also, it also generates some kind of behavior, okay? And then from here, this figure looks a bit funny, but the neural correlates of consciousness itself can be also, preceding neural correlates of the behavior. And behavior, I would actually count this verbal report or behavior report as a neural correlates of behavior. And then also other types of behavior. Here, their behavior means like eye movements or pupillary dilation or heartbeat or something like that. But these 3 components, if you distinguish conceptually, what we can do is to fix 2 while varying the only the rest of the 1 component, or fix 1 component while examining the relation of the 2. What I mean by that, so that's let's clarify that with the example. So here's a table 1 from their paper. The first table talks about empirical experimental paradigms, okay, in the normal subject, normal states, okay. And then here, the first one makes stimulation, NCS or NCB behavior remains constant, while NCC or subjective experience consciousness part changes, okay? And for example, in terms of perception, they suggested that we can study the neural correlates of binocular rivalry. We'll explain this kind of each of the phenomena as we go on, starting from this week and so on. And then also we can study neural correlates of episodic recall. So without changing stimulus or behavior, I can imagine, I can just recall what happened yesterday or what happened 2 days ago. And in fact, that visual imagery experiment for the vegetative patient that I covered in the early weeks is, in a sense, this type of the experiment. And then another possibility is this action, neural correlates of the awareness of intention. So here, you don't do anything. So the stimulus and the behavior remains constant, but You just try to think about or move about. Okay, and recently there was a very interesting study of the intentional internal speech, time locked with like counter like 5, 4, 3, 2, 1, and then you try to say something, but just internally. And we can find the ERP evidence of internally time locked speech, and the contents of that speech can be to some extent decoded as well. That's a very interesting paper. And then the stimulation changes while subjective experience remains constant. That's the opposite of what I discussed above, which could be the neural correlates of changes in stimulation without awareness. So we will go into this kind of paradigm in 2 weeks or so. One of the most famous example is change blindness. Even though the stimulus itself is changing, many of the subjects do not experience, there was a change. And some of the journal club presentation that you guys did in week 2 and 4 relates to this type. And also in terms of memory, neural correlates of unrecognized old items. So here you present different kind of stimulus. So the stimulus itself changes, but sometimes you see things that you saw before and then say that, no, I have never seen it, or the other side as well. So that's corresponding to or highly related to the situation of false alarm or miss in the signal detection task that I discussed a couple of weeks ago. So in that sense, this side is roughly speaking, focusing on the mistake that subjects believe that is correct with a metacognitive, mistake. Okay, and then in terms of action, neural correlates of stimulus eliciting action without awareness. So here, present some kind of priming stimulus or something like that. That you're not aware, but your behavior changes, and but you remain subjectively unaware of the stimulus. Again, some of your unconscious processing literature in the journal club is categorized along this category. Okay, and then behavior changes while subjective experience remains constant. This is correct guessing without awareness. That's a blindside situation, or correct correlates of the implicit learning. That's also similar to blindside, but you can make an unconscious learning within the normal subjects. And also neural correlates of implicit motor behavior. Again, like a blindside situation in the normal subject. And then the next one is abnormal situation, abnormal states, like the ones that are reported by the patients or under the hallucination or something like that. Okay. So here, let's go with the first one. Subjective experience changes, and then stimulation of behavior remains constant. And then in terms of percept, you have a situation of hallucination, as I said, and also memory, correlates of confabulation. So you don't have actual, events, but you feel like you had something. So deja vu would be something like this. And then action, abnormal intentions, delusions of control. So I might have mentioned this in the previous lecture, but there are cases of the Aryan hand syndrome, where patients report that your hand, her hand is moving regardless of her intention. We'll encounter some of these in later lecture in the split brain patient as well. And then another category, it would be stimulus changes, subjective experience remains constant. It's in a blind field in the blind side, and unrecognized items in amnesia. So here, amnesic patient who we'll cover in the later lecture, see the stimulus, and then they say that, no, I don't remember anything about this. However, they show some kind of behavior evidence to show that he actually likes this thing, and familiar face that he saw before, or show some aversions to things that were associated with, let's say, electrical shock and things like that. And then neural correlates of stimulus eliciting unintended actions, similar story. And then behavior changes, subjective experience remains constant, reaching from agnosia. So here, agnosia is a person who do not see the object regardless of their ability to reach to the objects and so on, which we'll cover again 2 weeks from now. And then implicit learning in amnesia patient, which we covered already. And then neural correlates of unintended actions, similar story again. So all these numbers here in this table, something like this, is actually the reference number of this paper. So if you find all these entry interesting, then I would recommend you to go into some of these papers and then use the Google Scholar to find what's the latest thing that is known about this neural correlate study, and then go from there for your final assessment. Okay. All right. So in summary, for this first part of the lecture, I introduced the concept of the NCC, Neural Correlates of Consciousness, and that's an empirical research program that has made a huge advance since 1990s. And Christoph Koch's review in 2016 brings updated notion of NCC. And there are many empirical approaches, as you saw in the last tables, some of which can be great inspiration for your final research proposal. Okay. So in the next part of the video, we'll go into the paradigm example of whether the NCC can be found in particular area of the brain, the B1. Okay, so the next next part of the video is going to talk about where is the NCC, and then specifically we ask as a B1 as a first target for the NCC. Based on our discussion about blind side, the natural candidate to ask whether it's NCC or not is the V1. Of course, the blind side patients were known for loss of phenomenology, and it's primarily due to the B1 damage. So let's see the evidence for and against it. To get to that, I want to first introduce the one psychologist's methodology, which is called the adaptation technique. And some people say that it's a psychologist's microelectrode. And here's the definition of the after effects or adaptation in Quest for Consciousness glossary. So it says a prolonged exposure to a stimulus attribute causes a short-lived deficit in the ability to detect that attribute. And here it mentions about orientation-dependent after effect, which we'll go into next. And in some cases, the opposite attribute is seen, as in the motion after effect, which again, I'm going to introduce in this lecture, where the observer sees upward motion after being habituated to downward motion, also known as waterfall illusion or color after images. Okay, so after effects are thought to be caused by a recalibration or adaptation of the underlying neurons. So let's see what it means. Adaptation can cause a vivid alteration of conscious phenomenology without changing the stimulus. So this is actually a good definition for the fixed neural correlates of stimulation while varying the neural correlates of consciousness. If the adaptation is caused by specific neurons, then it can be used to argue or infer where in the brain the adaptation is happening. And adaptation can be combined with independent manipulation of consciousness or visibility from other kind of processes, such as attention or report and memory or expectation. So that's very important kind of tools for consciousness study. Okay. Okay, so here's the first example of After Effects. If you first focus on the central cross, okay, and then if you here you shouldn't move your eyes and also make blink here, and then just look at the center. Okay, are you ready to see the after image here? Okay, all right. So 3, 2, 1. I hope that you see you saw the image, colored image on each of the corresponding gray square region. And now probably your after image is completely washed out, so that you can compare, confirm that nothing is there actually. But if I go back and forth like this, then without moving your eyes from the center, you can see that color of the image is quite vivid, and then but there is nothing physically presented here in this gray square. Okay, so this is one form of the after effects called after image, negative after image. And this is believed to be caused at the fatigue of the retinal neurons, but there are important studies that we will go into the details in next 2 weeks that shows that it can't be the case. All right, and that it can't be the case that the purely after images are arising from the retina. Okay, so then now I want to also introduce this very interesting website that lists lots of visual illusions that you can see. This is collected by this Professor Michael Bach, and you can see lots of icons that represents many of these visual illusions. And so if you have time, I would encourage you to go through these illusions to learn more about visual illusions and the consciousness. But in particular, we are going to look at this lilac chaser. So here, this is a very strong case of after image combined with the troxler fading and also other things. And you can change the parameter here as you wish. So what you have to do is to fix it at the center and then just observe what happens. Okay, don't move your eyes and don't blink, but just stay focused at the center. And if you're like me, then what you'll notice is that instead of seeing a bunch of pink circles, I started to see green blob going around, and then after the green, all these pinks are erased from consciousness. So this is a very striking kind of illusion where what you see, green thing, is not at all on the image, but what's there, a pink thing, is not present here. And then if it's difficult for you to see it, then we can lower the saturation like this, and then do it again. And if you're still having a difficulty in these illusions, and close one of your eyes, and then you will probably see that only green is going around, and then no pink is visible. Okay, so this is a combination of after image and troxler fade. Okay. So the next one is another types of the illusion, and this is something that I made by adjusting this stimulus. Here you will see a bunch of horizontal grating, faint grating on the left side, and the vertical grating on the right side. And then what you will do, what I'll do is not to ask you to look at the center, okay, center white circle, and then don't blink or move your eyes, and then just stay there, okay? And now I'm alternating between black and white pattern of this grating, so that it is complementary to each other. In this case, the after image should be not generated because of this white and black pattern. Okay, and then usually for this type of the after effects, not the after image, it takes roughly like 30 seconds to 1 minute to make the effect most strong. And then it goes, let's go to the test image briefly and then come back to this, okay? Tell me what you see on the left side and the right side. 3, 2, 1, 0. Okay, one more time. 3, 2, 1, 0. 3, 2, 1, 0. Okay, if it doesn't work, you can try it again by just using class website Google Slide. But what I hope you to see is that the left side horizontal grating become probably blank, and then vertical ones remain there. And if that works, that means that momentarily your vision sensitivity to the left side of the vision became very blunt on the horizontal grating or lines. And on the other hand, the right side became more sensitive to the vertical grating. So this is the adaptation, and can be explained by the fatigue of the neurons in, for example, V1. As I said in the last week, V1 neurons contain responsibility to the particular orientation, and those neurons that were repeatedly stimulated by this horizontal stimulus should be fatigued, and therefore it can't generate a strong response to the another horizontal stimulus on the left side, whereas the vertical ones were more or less remaining intact. So you see the vertical one on the right side very, very vividly. Okay, so why I'm talking about all these adaptations, the reason is the following. So orientation dependent adaptation can serve as a probe for conscious experience and considered to occur in V1. And then, as I said, the neurons in the retina and the LGN do not have possibility to generate this after effects. And then the question is, in response to consciousness research, is that do we need to consciously see the adapter during the adaptation? So you may find it weird, but here's the clever technique for addressing this question. Okay. All right, so if I ask you to stay fixated at the center, cross here, and then without moving eyes, can you see the orientation of the grating from next, second from the rightmost grating, like this one? It's either right tilted or left tilted. And then you can actually see it now at the foveation, and then you see confirming that, oh, this is actually tilted to the right. And then coming back here, and then coming back to the fixation cross, and then see whether you can actually tell the difference. Most likely no. This is called a crowding. If the objects is surrounded by similar object before and after, the visibility of this surrounded pattern becomes lower. So that's the crowding phenomena. And it's not a matter of the eccentricity. So you can see if it's presented in isolation, then now you can fixate on the cross and then tell me that, oh, it's actually tilted to the right. This is very easy. And then again, you do it, then it's, even if you know it, you can't tell, okay? And then the idea of this experiment is to see the difference in the sensitivity for the same orientation, like this, or as adapted orientation, or the different one, like this one. And if the orientation dependent effect exists, then the visibility of the same grating will be lower, or threshold to be higher, and then the different one to be more visible. So this is what they found. So here on the left side, this is when the stimulus was in the single condition. So this is all visible situation, okay, single. And then the contrast threshold elevation. So that you need 6 times stronger stimulus to see to make a correct discrimination if the direction is on the same, orientation is the same. And if it's different, then there is some kind of enhancement, but it's not as dramatic. And in the case of the crowded situation, regardless of the fact that you didn't see the stimulus, the contrast elevation is observed, like here. And it's slightly lower, but still much significant. And what it means for our purpose is that the crowded and invisible orientation still evokes strong threshold elevation. And if it is generated by the V1 neurons, then this invisible orientation does not contribute to our conscious perception of the orientation. That's the logic, okay? So not only that, Quest for Consciousness, chapter 6, contains several evidence in favor of the V1 as not the site for NCC. One is the dreaming study. As some of you presented or will present in a journal club, during the dreaming, we can record the neural activity. And then the famous example, famous result is that during the REM sleep, where people dream a lot, V1 tends to be suppressed. And whether suppression corresponds to non-conscious is a matter of the debate, but that's one evidence. Secondly, that the V1 neurons property do not follow the property of consciousness. For example, as I mentioned briefly, within layer 4 of the V1, neurons respond only to the input from one eye, but not the other. So the eye of origin information is present in the V1, especially layer 4. However, if you present the stimulus, maybe if you have an access to the virtual reality goggle, you can do it. If you present the stimulus through the right eye or through the left eye, you can't actually make a above chance discrimination. And this can be taken as an evidence that the eye of origin information is not possible to consciously access, okay? And if the V1 information is something that we can be consciously accessible, then at least this is inconsistent. That's the idea. The another prominent evidence is that V1 neurons, if you record it from there, it shows a very strong influence of the blinks and also eye movements. Every time when you blink, again, it's almost like a saccadic suppression, but start before the blink and after the blink, there is a time of this sensitivity drop. And also, you do not see the word, by default, blank, right? You can actually close eyes for a while and then see, that's how you see. And then the duration of the blink can be 100 to 200 milliseconds each time. And we tend to blink. This has an individual difference, but once to twice, maybe 3 times per second. But we don't rarely, perceive the blinks related to blanking. But V1 neurons do show the time-locked, activation or deactivation induced by the blinks, as well as eye movements. So that's also in favor of the hypothesis that V1 neuron activity is not really correlated with the consciousness. And as a result of this thing, and also Crick and Koch in 1995 presented this so-called V1 hypothesis, saying that light comes into the retina and then retina to LGN and V1, but V1 also does not have a direct connection to any of this prefrontal or premotor cortical areas. So this one, this anatomical connection does not exist in, at least in monkeys and humans, most likely. And there, they thought, hypothesized that conscious experience should be related to some kind of motor output. And if there is no direct connection, that means that most likely it remains private and not something that we can be conscious of directly. And this is a kind of the conjecture that I don't believe right now, but it was received at that time. Okay. And so this is what I said. V1 doesn't have direct projection to the prefrontal and premotor cortical areas. So in summary, this part of the lecture presented the Crick and Koch V1 hypothesis, and that promoted various empirical research since 1995, not only summarizing what we knew by that time, it also promoted tests of whether V1 neurons activity is correlated with the conscious experience in various situations, and it still continues. And V1 involvement for the reported or reportable percepts are unlikely at this moment to be based on the correlated with V1 activity based on various perceptual and neuronal findings. Some of these will be presented in a journal club in week 2, 4, or 8. However, in terms of its involvement for consciousness per se, such as full MCC or the feeling of the space, this is still unclear. It's also plausible that V1 is contributing to consciousness directly in a way that we cannot really verbalize, such as a texture or distance or size of the things. Okay. So in the next part of the video, we are going to talk about what about the other areas in terms of the candidate for the NCC beyond the V1. Okay, so now we are going to talk about the visual consciousness and visual areas beyond the V1. So far, I have been mainly explained about the visual system through eyes here, and then that through the optic nerves goes to LGN here, and then this bundle of the axons is called optic radiation, and then that reaches to the back of the head here. That's a V1 here. And then the upper part of the V1, which corresponds to the lower visual field, connects to V2 areas that surrounds V1, and then V3. And then that's surrounded by something like V3 and V7. From here, it becomes more complicated, and so on. And on the bottom part that goes below this surface, that goes to B2 and B3 as well for the ventral areas, and then B4 and BA. And then also the important architecture is this MT or B5, that's we are going to talk about today. And then, yeah, so that's the overview of the visual areas beyond the V1. So in terms of understanding the visual cortex, it is important to understand what the cortical flattening is and how the retinotopy looks like for V1 to V4. So to explain this figure, I think it's first better to look at the movie, such as this one. Okay, so this movie basically explains how the modern neuroscience actually analyze this brain data. So this is an image taken from MRI, and then initially it removes all these non-cortical surface areas,
and computationally, and then it tries to inflate as if it is sort of the balloon, and then so you can do this by computational kind of analysis using a technique called topology, and then this wrinkle, it gets completely inflated like this. And then at this point, you can cut the brain in a way that that can unfold onto the two-dimensional surface. And that's how we see these types of the retinopathy image to be seen. Okay, so here's after the cortical flattening of the presentation of the right hemifield, right cortex for both macaque and human brain. So here's the key for the visual field. This is a left visual field, okay? And as I have already mentioned, the left visual field goes to the right hemisphere. And then the upper part, so the key to this location is the fovea. Fovea gets projected onto this circle, okay, here. And also other areas also have the fovea representation for both monkey and for human. And then the upper part of the left visual field goes into the lower part of the visual cortex. So here, this is actually, in a sense, a bit difficult to imagine, but the right cortex is now cut around here and then opened it up for B1 and B1. So this lower part gets here and the upper part of the B1 gets here. And that looks like, so the most upper part comes in this area, and the most lower part goes into the dorsal upper part of the brain, okay? And then same goes for the macaque. And then instead of going to the right visual field, it flips to next visual field in B2 in both human and macaque. And then B2 continues from the upper representation for the ventral part of the B2. And then lower part of the vision goes to dorsal part of the B2, and so on. And then B4 is located only for the ventral side. And then B3A corresponds to the dorsal side. These functional anatomical name can change over the time, so you don't need to worry too much about it beyond B2, B3, B4. Okay, and then so all these ventral side versus dorsal side has a sort of mirror symmetric kind of representation, but only within right hemisphere for this presentation. And this island of MT is the part that I'm going to talk about towards the end of this lecture. The B2 neurons are known to respond strongly to the illusory contours or illusory lines. So if the monkey is fixating at this, kind of central cross, and then you move the bars, then if that matches with the receptive field of this neuron, then it neurons fires like drrr, drrr, very, very strong, response when it matches with the receptive field. But when again neuron is fixating here, if there is a line like this, then you can kind of infer that there's a line going on. So when you move like this, there's a little bit of the response, and then, okay? So different rows are different repetitions, typically. But if you insert a little bit of that space between this location, here, there was no space, then your inference about this, line occluded by this black part becomes much, much weaker, both cognitively and visually. And that mimics this B2's silence of the response here. On the other hand, if you make some kind of the lines like this, composed of the many bunch of the lines which is slightly mislocated, then you strongly feel like there is a line here. And that produces a very strong response in B2. Taking advantage of this type of the finding, so there were some researchers that utilized this knowledge for the, again, consciousness experiment as follows. So here, what the researcher did is to use the illusory line to generate an adaptation and a crowding experiment, like the one that I explained a couple of minutes ago. So here, they asked the people to look at these red dots, and then across this goes over the time. So over the time, they generated different bunch of the stimulus here, but important thing is that the second from the top here always generate illusory horizontal line. And then they ask people to adapt to this display for, let's say, 1 minute or so, and then they showed some kind of flash to indicate that this adaptation period is finished, and then tested with the visibility of this horizontal or vertical adapted or non-adapted stimulus. And then when they did the important check of the visibility of the stimulus itself during the crowded situation, all the subjects showed chance performance. Okay, that's very important to check. And then next, when they did the adaptation task, they found, as expected, if it's tested with a different stimulus, like here, then the performance was very accurate, in terms of the detection. On the other hand, if it's the same as adaptation direction, then orientation, then they were near chance, but still above chance. And the important twist for this particular experiment was that they added an attention manipulation, actually, because they knew that this kind of adaptation should happen without visibility. They added this important experiment. So asking subjects to detect the color, detect a number that is, for example, even and black or something like that. And then this number, if it changes very, very quickly, you need to really pay attention to this, number stream. And then if you do that, then your attention to these, adapted orientation, should be weaker. That's their hypothesis. And then when they did that, then this differentiated orientation dependent adaptation effect of the invisible stimulus became, pretty much gone. This is a bit complicated logic, but here's what they did, is a summary. Okay, so first of all, the neurons in B2 seems to respond to illusory lines, and that has been replicated a lot and across monkeys and humans using fMRI. And then also illusory lines can generate adaptation effect, even though they are not visible based on the crowding. However, this non-conscious adaptation effect requires attentional amplification. And here, you are not really trying to see it by attention. I mean, if you try to see it, you can't see it. That's something that you can also try with the demonstration, a couple of the last video by yourself. This case, attention is just availability to give some kind of, further processing. But just withdrawing that attentional resource makes this non-conscious adaptation impossible. So non-conscious processing does require attentional amplification in this case. Then this poses a question about sufficiency of attention for conscious perception. already, in week 2, some of you have raised a question during the tutorial that, is attention necessary or sufficient for consciousness? And here's one particular example where unconscious processing requires attention. And the opposite way of saying is that attending something does not guarantee that you see things. And here's the next, in 2 weeks, we'll go into the sufficiency and necessity of attention for conscious vision. Okay. And in the next video, we'll go into the strong candidate for the NCC for visual motion in MT. Okay, so the last part of this video, this week's lecture is about visual motion perception and the middle temporal area. Sometimes it's called MT, sometimes V5, depending on where you are. In US, it tends to be called MT, and in UK, it's called tends to be called V5. So the V1, V2, V3, that's what we already covered, and then MT or V5 locates slightly anterior or temporal to the visual cortex, like this way. And this area tends to contain lots of neurons that respond to the visual motion. And just one word of caution, we talk about MTL in the at least in the next lecture, and it is a medial temporal lobe. Okay, medial means the inside compared to lateral, and the temporal means this temporal lobe. And middle temporal lobe, MT, is the sort of the middle of the temporal lobe. So that's a different medial temporal versus middle temporal. Okay, and the medial temporal lobe contains structures such as the hippocampus and the amygdala that are important for memory or emotion. So don't confuse MT and MTL. Okay, so let's go to MT. Before getting to the MT, I first want to show another video, which is about the motion after effects and its relation to consciousness. Okay, and again, we go to this Michael Bach's website, and this one is about rotation motion. So here, I want you to fixate on the center part of the video, and then do not move your eyes. Okay, this blue-red part. And if you are like me, and then you can do it again. Don't move your eyes and don't blink. Adapt, adapt, adapt. And then after a while, you see this, so this Buddha thing, which is located probably in Kamakura in Japan, that should appear expanding to you. And then again, once again, just in case you don't see it. Yeah, and it expanded. So this motion after effects, like other after effects, orientation after effects or after image, it accumulates over the time. So the more you do it, you start to see the expansion much stronger. So the next time the expansion should be much stronger. Yeah. I feel like radiation kind of line coming out from the Buddha as well. Okay, so that's the motion after effect. In this case, it's a radial motion after effects. Okay. So we can do the same kind of experiment as the crowded version or illusory contour version for this experiment. That's what people have done. This is a paper by my previous postdoc, Lisandro Kaunet. He did it not with me, but published in 2011. So the point of this paper was that the adaptation to the visible versus invisible motion was compared as a function of the motion's complexity, simple version versus complex version. The first experiment, what he did was to present some kind of grating to the non-dominant eye. Most likely, many people have non-dominant eye on the left eye. And then to the dominant eye, he presented some kind of flickering pattern called the continuous flash suppression, which I'll explain in later weeks. And that's something that I discovered when I was a PhD. So here, what he did was to present either this grating that moves either only upward or downward, and then for roughly 1 minute, and then presented another stimulus right after that. And then after that, as I said, this kind of motion after effects accumulates, but after once it accumulates, it stays on. So 2 seconds of the top up adaptation and then test, top up, test, top up, test, top up, test kind of thing. So under the visible condition, you don't show any kind of continuous suppression, and then just adapt to this, motion for unidirection. If it's up condition, up for 1 minute, and then test, and then visibility for upward or downward, and then go to the 2 seconds of the up, and then up or down kind of thing. And he tested it with both eyes, adapted eye or non-adapted eye, like this way. And he crossed it with visibility suppression. So if he didn't show anything like this, then this one becomes visible. But if she showed the continuous flash suppression, he was able to suppress the stimulus quite effectively. Okay, so that's the first important condition. And then next, what Lisandro did was to present complex motion. So here it looks like sort of the expanding or contracting motion for the Buddha demo that I showed, but he used a spiral motion. And the spiral is believed to be processed in 1 step higher than the MT. It's called the MST, middle superior temporal sulcus. Okay, temporal lobe or something like that. And then here, this next stage of the MT, it processes a radial motion or spiral motion. And he made again visible or invisible kind of condition, and the test was whether the contraction or expansion of the spiral was perceived. And then importantly, what he did also was that he added another attentional condition. So importantly, what he added was this attentional condition. So on the dominant eye, suppressive stimulus was presented. He presented some kind of letters similar to the attentional manipulation in the previous study. There, people were either attending to these letters or not. And then he asked the question of whether attention or conscious visibility is necessary to get the motion after effect. So here is the result of his finding for the simple motion. So in the case of the simple motion, when the stimulus was visible, so this is visible, and then this black one is invisible. No attention task or easy attention task or difficult attention task. So it's a 2x3 design. As long as stimulus was visible, no matter what attention was withdrawn, stimulus was processed and the motion after effect was very strong. And there is some kind of relation as a function of the strength of the attentional distraction. But importantly, invisible stimulus still produced lots of after effects. And it's strongly above chance. Above chance performance is defined above 0.5 here, okay? And then the complex motion. So in the case of the spiral motion, when he did it with the seeing adapter, no suppression, then the adaptation was very strong, and regardless of the again attention. But if it's invisible stimulus, then even the attention task was very weak. It already became at chance level, and this one as well. So the visible and the complex motion evokes motion after effects, but invisible complex motion evokes very weak motion after effects, if anything. And this kind of psychophysics experiment has been also combined or backed up by the fMRI experiment in humans. So here, it was one of the first studies on this kind by Roger Tutel in Massachusetts. What he did was first to locate the area MT in this inflated brain, or it's embedded sometimes in subjects from the right visual field. And then he asked people to adapt to this expanding motion for roughly 40 seconds or so, okay, 40 seconds, or a contraction or expansion. And then after that, he put the stimulus on for 40 seconds again, and then looked at, invoked the motion after effects from the subjects. So here, 40 seconds of expansion, and then 40 seconds of the static, and then expansion or contraction, and then static, contraction, static, expansion or contraction, and then static. So that's the sort of the design of the experiment. And each of the block is 40 seconds. And then this is an fMRI response at the, this MT area. Okay, so MT area's response was like, at the beginning, it was baseline, and then it increases, and then goes down, but with substantial sort of, residual activity here. And that's what Tutel analyzed further on, combining with the psychophysics. So what they found was that aligning across all these trials, they found that at the end of the motion, when the stimulus was presented, then stationary stimulus was presented, then there is strong effects of transient increase in fMRI in the MT areas, okay? And that invokes this stimulus. And this one is already subtracting this motion component already. And then the important thing is this, quantification of the psychophysical motion after effects. At different time point, they quantified how strong the people were experiencing the motion after effects. And then what they found was that this motion after effects decay. And that's something that you probably also feel if you test this, Buddha, motion after effects thing by yourself. After the time, it starts to stop. So this is a very nice correlate it's quite a tight correlate, I should say, between the motion after effect or perception of the visual motion, quality of the visual motion, and neuronal activity. And importantly, this type of the correlation should be also compared with the causal evidence. And in terms of the human, most straightforward causal evidence comes from the patients. And here, the patients receive undergone MT, bilateral MT lesion, reported this condition called akinetopsia. And this is a specific deficit in the perception of the visual motion, but nothing else due to the cortical lesion in and around the MT area. And this type of the phenomena has been reported in the monkey. Like, blind side case, physiologist has made a lesion in monkey and then characterized the performance, which looked similar to these MT patients, basically. And this video is fiction, but I find it very compelling. So let's watch about that and then see.
43-year-old woman was admitted to a hospital in a stupor. She had suffered for 3 days with severe headaches and vomiting. Several tests run then and later suggested that a stroke had damaged both sides of her brain at the lateral border between the temporal and occipital lobes, here indicated by a dark spot. 19 months later, in May 1980, she was examined by neurologist Joseph Zill, and in most respects was found to be normal. She could read, write, and calculate. She had no trouble moving her body. But, as Zill reported in 1983, she had an unusual complaint.
The visual disorder complained of by the patient was a loss of movement vision in all 3 dimensions. She had difficulty, for example, in pouring tea or coffee into a cup because the fluid appeared to be frozen like a glacier. In addition, she could not stop pouring at the right time since she was unable to perceive the movement in the cup when the fluid rose. Furthermore, the patient complained of difficulties in following a dialogue because she could not see the movements of the face and especially the mouth of the speaker. In a room where more than 200 people were walking, she felt insecure and unwell and usually left the room immediately because
people will suddenly hear all their, but I did not see them moving.
So
she could not cross the street.
So that's probably enough to get some kind of intuition about what the patient would have experienced.
Okay, so that was a fiction, but it looks kind of comparing description of the empty lesion patient. Sometimes it is described as a strobe-like situation. But it's a very rare lesion, so we don't know much about this type of the patients. It needs to be studied further. All right, so then in terms of the columnar organization, I mentioned it briefly last week, but MT contains neurons with the columnar organization across the direction and also depth of the motion as well. And this means that it's actually useful for us to think about the sort of micro-stimulation experiment that I'm going to talk about in next slides, because electrical currents that is passed in a local neighborhood of the neurons, which prefers that vertical orientation, vertical direction of motion or something like that, then many neurons that prefer these direction can be effectively stimulated by local stimulation. But before going to the microstimulation, I need to talk about this very famous paradigm of the MT neuron and psychophysics. So this is the stimulus called random dot kinematogram, pioneered by this motion physiologist Kenneth Britton and also Michael Shadran and also Bill Newsom. As you can see, so this the degree of the coherence or correlation of the motion can be parametrically manipulated like this way. So 100% correlation motion means that all the dot moves in the same direction, and no correlation means that from one frame to the next, all the dots are redrawn completely in a different location. So it looks really a random motion. And 50% correlation means that 50% of the dots are drawn in the random location. So how it looks like. Okay, so here's the example of the random dot motion stimulus. So if you stop the frame, you will see that bunch of dots, and then initially it's moving in a completely random, redrawn in a different direction, location, and so you presumably didn't have
competing, motion direction. But if most of the new dots move to the left, then everybody see the motion to the left. Okay, so nice thing about this dot motion stimulus is that you can change the number of the dot moves into particular direction and also the other direction in a parametric manner. So this groundbreaking study conducted by Bill Newsom and colleagues basically showed this motion to the location of the receptive field of the neurons in MT. And then they presented different level of the coherence motion to the monkey, okay, at the location. And then they also asked the monkey, trained the monkey for months and so on, and then let them report which way the motion was moving. In this case, let's say upwards or downwards. Okay, and then when the motion stimulus contained quite a bit of the strong correlation, let's say 12% or so, then one single MT neuron may fire a lot, let's say up to 100 hertz, 100 spikes per trial. Okay, spikes. They counted the number of the spikes. And here, so this is a 12.8% trial, and then some of the trials produced this neuron fired a lot. On the other hand, if it's let's say in this case it's upward motion, and this is a downward motion, then this neuron didn't like this downward motion. So sometimes, most of the cases, they fired much less. And then there is a mean peak here, and then here peak as well. And you can imagine that we can do the signal detection type analysis by putting the criterion here and then compute the area under the curve. And that's what they did, in fact. And then they generated a different level of the coherence, and this 0.8 is almost invisible to make upper or lower type discrimination. And as the coherence goes down, correlation goes down, neuron gets confused. That's the upshot of this figure. Okay, and then what was interesting is that when they look at the monkey's performance, which is this white dots, this is monkey's report. So remember that during all these experiments, monkey was also reporting this percent, and then they plotted the proportion of the correct as a function of the correlation. Okay, the stronger the stimulus, the monkey became much accurate. Okay. And then this is one particular exemplar MT neuron. So performance by coming from this, they put the criterion here and then said, okay, so this one corresponds to, let's say, 80% correct. So it would put something like here, or maybe because it's a 12%, it corresponds to something like this, almost 100% accuracy. And then if it's a correlation of 0.8 here, then it's almost indistinguishable, so it corresponds to here, and so on. Then they found that on average, they could always find a neuron that responds at least slightly better than monkey. And MT is a bit farther away from the motor cortex, which controls the monkey's report. They thought that there is some kind of noise coming in and degrades this purely motion information down the way to so eventually will result into monkey's report. So far it's it's a great study, but not super relevant for consciousness per se. But a further experiment by Zaltzman did more relevant experiment. So here the monkey was fixating at this location, and then they made a decision between this direction of motion and this direction of motion for one particular MT neuron. And then monkey was reporting the direction of motion by the eye position. If it's moving this way, then they move the eye here. Then if it's moving this way, then move the eye here, something like that. And then not only the visual stimulus was presented between T2 to T3, they also delivered the electrical stimulation around the neuron that they recorded after the recording of the neurons. Okay, and then they found that as a function of the correlation, without the stimulation, okay, monkey reported something like this way, and then proportion of the preferred decisions for this particular location. And this is a 0. So, at the 0, then monkey tend to say, 50-50 in this case. And then there was some kind of bias to say, I say, to the bottom or something like that for this particular experimental session. But the important thing is that when they deliver the electrical stimulation, microstimulation, then the monkey's report, conscious report, became more biased towards the preferred direction. So the stimulation increased the proportion of the report by roughly 10% to 20%. But that corresponds to, so this is actually quite significant because here the electrical stimulation skipped all the way from the retina to LGN to V1 and then directly stimulated MT. And this artificial stimulation was, in a sense, confused or perceived by the monkey, almost like a motion stimulus. And then that's why they reported like this. Okay, so this motion electrical stimulation experiment is very important in terms of the building this causality between the MT neurons activation and motion perception. So in summary, MT's causal role for the visual motion qualia is strongly implicated by the phenomenology and psychophysics and the fMRI and lesion and the neurophysiology and microstimulation. It's very compelling. And bilateral lesion abolishes motion qualia. This is very important. And also microstimulation causally interfere with the motion. So these two things are one of the most compelling evidence. Now, important question for the consciousness researcher is why does MT generate motion qualia? And at this point, really nobody knows. And you need to think about what is special about MT. Okay? And some may say that it is an input, or some say maybe it's the output. It is directly related to the behavior or sort of the verbal report of seeing motion. But that doesn't make much sense. If you don't report anything, but you can still see the motion. And in terms of input side, if it's the input, let's say retina to MT in a certain configuration generates this pattern of the input, and that defines MT's role of visual processing and then generate quality. Why this input actually needs to cause this motion qualia? That's the question. Another possible answer would be there may be something really specific about MT's neurons biophysics. Maybe some special receptor that generates motion sensation, which we can't really rule out at this point. And also another likely scenario that at least what I think is that in local neurocircuitry, which is related to all these layer structures and also neurons excitatory and inhibitory population, and also columnar organization, all of these are potentially related to the key to the explanation of why MT generates motion qualia. And this is exactly the hard problem. And if you can figure this out, it's potentially like a Nobel laureate prize. It's really unclear at the moment. And the philosophers and neuroscientists are all at a loss. And then so that's another form of the hard problem. So that's another form of the hard problem, which we will further discuss and also look at by looking at the other areas of the brain as well. Okay, see you next week.